Thanks for coming to my talk. So in mammalian species, maternal nurturing behavior, such as food sharing, playing, or attention to their offspring, is a vital component of maternal investment, with higher rates of nurturing associated with the development of a number of preferential offspring phenotypes, such as improved social cognition or appropriate physiological responses to social stressors and cues. Given these adaptive benefits of nurturing, we might expect mothers to always be highly attentive of their offspring. However, we know that in many mammal species, including humans, there are clear inter-individual differences in maternal nurturing behavior, so-called maternal styles, with some mothers consistently more nurturing and attentive than others. Where this variation comes from is key to understanding the selective pressures shaping the evolution of different maternal care patterns among mammals. So the terminal investment hypothesis proposes that mothers should invest more in their offspring as the mother's age and her future reproductive opportunities become more limited. Meanwhile, the Trivers-Willard hypothesis proposes investment is predicted by the interaction between maternal condition and offspring sex. So when mothers are in good condition, they should invest in offspring in which she can have a large impact on competitive ability of that offspring and thus maximize her own inclusive fitness. So in chimps and many mammals, this would mean investing more in sons when in a good condition and more in daughters when in a poor condition. Although both of these hypotheses have received support from studies in a number of mammals using interbirth intervals as a proxy measure of maternal investment, it remains to be tested whether these predictions apply when we consider nurturing behavior as the specific form of maternal investment. So in our study, we examined whether either of these ultimate level hypotheses explain differences in maternal nurturing. And then to try and get a more holistic understanding of variation in nurturing, we also considered a key proximate mechanism, namely oxytocin secretion. So this neuropeptide is produced in the hypothalamus, and elevations in oxytocin are correlated with the timing of maternal nurturing in rodents, sheep, humans, and other primates. However, whether inter-individual differences in oxytocin co-vary with nurturing styles is yet to be tested in a long-lived mammal or in a wild animal system. So to do all this, we conducted our study on the chimps of the Thai chimpanzee project in Côte d'Ivoire. We had data from 30 mothers, and we had around 2,500 hours of behavioral data to work with, and over 300 urinary oxytocin measures, with all this data coming from around 5 years of data collection. To analyze variation in nurturing behavior, rather than aggregating our data in the form of time budget analyses, we instead opted to use Bayesian categorical models. So these allow us to estimate the probability of certain behaviors relative to a reference behavior. So you can think of the outputs as similar to a Bernoulli model, but with multiple outcomes rather than just being a binary response. To do this, we divided our maternal behavior into 10-minute intervals, recording the social choices made by mothers within those intervals. This generated a data set of close to 15,000 choices, around 500 per mother. We categorized social choices within these intervals as either nutritional nurturing, so any nursing or food sharing with their offspring, socio-positive nurturing, any grooming, playing, or affiliative attention towards their youngest offspring. And then to account for the general sociability of the mothers, we also included as a category whether the mother had any positive social interactions with individuals other than her youngest offspring during the interval. Our reference category was non-social, so behavioral intervals when mothers engaged in no positive social interactions. So from this model, we found a clear effect of offspring age on the social choices of mothers. So in this plot on the y-axis, you have the probability of a particular social choice relative to being non-social, and on the x-axis, you have offspring age. The colored lines represent draws from the posterior, colored for the different social choices. So the greenish color here represents the probability of nutritional nurturing, the blue, the probability of socio-positive nurturing, and the purple is the probability of socializing with individuals not the youngest offspring. And hopefully what you can see is that the probability of both nurturing behaviors increased with the age of the offspring. And this was also associated with the decline in social interactions with other individuals. Nurturing behavior appeared to peak between the ages of 2 to 4 years old, which coincides with the weaning process in this species, after which mothers became increasingly likely to socialize with other individuals. Maternal age had the opposite effect of what we expected. So while maternal age had no effect on the probability of nutritional nurturing, we saw moderate declines in the probability of socio-positive nurturing as mothers aged. And this may represent some form of energetic constraint on the mothers as they get older. However, it's important to note that while we had strong support for the offspring age effects, the maternal age effects were rather weakly supported in our models. In addition, we found no support for the Travers-Willard hypothesis, and overall, we would conclude that maternal life history and related hypotheses are not particularly strong predictors of nurturing behavior in this species. So how did oxytocin relate to these behavioral patterns? Well, to test this, we fit Gaussian mixed models with urinary oxytocin concentrations as a response. So urine samples are easy and non-invasive to collect in wild chimpanzees, and we collected over 300 samples for this particular study, which were then analyzed using enzyme immunoassays to quantify the urinary oxytocin concentrations. And from the work in our lab and others, we find clear elevations in urinary oxytocin concentrations following cooperative and sociopositive interactions in chimpanzees and other primate species, indicating that we're able to pick up on meaningful changes in oxytocin levels using these urinary measures. So from our model, when we looked at the effect of offspring age on a mother's urinary oxytocin concentrations, we found a pattern strikingly similar to the relationship observed with nurturing behavior and offspring age. So on the left here, you have the oxytocin results with oxytocin concentration on the y-axis, and then on the right, you have the nurturing behavior model results once again. And hopefully you can see that maternal oxytocin levels increase with the age of the offspring, peaking between the age of 3 and 4 years old before declining. And this decline in oxytocin levels occurs at a similar point in offspring ontogeny to the behavioral nurturing decline. When we looked at how nurturing styles were related to oxytocin levels, we found that mothers that were more likely to nutritionally nurture their offspring tended to have high urinary oxytocin levels. So here we found a clear correlation between nurturing styles and oxytocin levels in this species. So to put this all together briefly, life history hypotheses regarding maternal investment were poorly supported when we considered nurturing behavior. Instead, the age of the offspring was the main predictor of nurturing behavior, and oxytocin levels co-varied with offspring age and nurturing behavior. Importantly, we also found that oxytocin levels were related to nurturing styles that were independent of offspring age effects or maternal life history. And with that, I'd like to thank all my collaborators on this project in the lab and in the field. And I'd like to thank you again for your attention and coming along. Thanks a lot.